Hello student, welcome back to Arunava bio class. So today we discuss about the very important topic of the cell for our NEET section. And last time we discussed about the cells. So at first we discussed what is the introduction about the cell, what is the cell, like what is the definition of the cell. Next we discuss about the different scientists and their contribution, like the Robert Hooke, like the Anthony van Leeuwenhoek. Next we discuss about the different types of microscope, like the light microscope and the electron microscope is there. Next we discuss the very important topic, that is the cell theory. Slated as one given theory, and after that Rudolf Virchow ultimately modified this theory, right? So this is the cell theory. Next we discuss about the overview of the structure of the cell. You know that ultimately all the cell contain the cytoplasm and the ribosome, that is applicable in both prokaryote and eukaryotes. That is applicable in both plant and animal cells. Very important some information in this picture from NCERT. It is very important, like what is the name of the smallest cell, what is the name of the largest isolated single cell. All of this information is very important for your NEET purpose. Like you know, the cell is a different shape, like the some cells are disc-like shape, columnar shapes, right? This is the different cells, and the example is given in the NCERT book. That is very important. Next we discuss about the structure of the prokaryotic cells. What is the prokaryotic cell? Like the bacterial cells, like the mycoplasma, like the blue-green algae, all of belongs to prokaryotes. There we are classified the bacteria according to their shape, like the bacillus, like the coccus, like this type of differentiation is there. Then we discuss about the structure of the bacterial cells, like the cell valvula, mesosome, nucleoid, flagella, pili and fimbriae, ribosome, and all the inclusions bodies. That is most important part. Cell envelope, it is have the three part: glycocalyx, cell wall, and the plasma membrane. And cell wall is, you know, made up of peptidoglycans, and the plasma membrane or the cell membrane made up of phospholipids. Next of one we discuss that according to Gram staining, which is scientist Christian Gram developed this type of staining techniques. We classify the bacteria in the Gram positive and in the Gram negative part. Escherichia coli, these bacteria belongs to Gram negative. On the other hand, Salmonella typhi ultimately also example of Gram positive bacteria. You know, next we discuss about the structure of mesosome. That is the invagination of the cell membrane, right? This is helping the bacterial respiration. All of these things we discussed in last chapter. And in the nucleoid, the DNA material of bacterial cells, like the flagella, help in the bacterial motility. Pili or pili, that is the sex pili, they are helping the bacterial conjugation process. Ribosome, that is the 70th type of ribosome is there. Maybe sometimes they are from the polyribosome, some inclusions, bodies or dead bodies present in the bacterial cell. After that, we come to the eukaryotic cell. So at first we discuss their overview. After that, you know that like the amoeboid cells, like the fungal cell, like the plant cell or animal cell, all cells are the eukaryotes or eukaryotic cell. Why? Because their nucleus are membrane bound. They are developed, right? And the membrane bound cell organelles also be present. And in the last class we discussed about the cell membrane and cell wall, and today we describe the structure, the endomembrane system, mitochondria, plastids, and the ribosome. And next class we discuss about cytoskeleton, cilia flagella, centrosome and centriole, nucleus, and microbodies. So last time we discussed about cell membrane, right? Cell membrane, they have very unique structural format is there in all the cells. It is present in the plant cell, it is present in the animal cell, it is present in bacterial cell. All the cells have the cell membrane. But then main chemical composition is that they contain the phospholipids, right? But the scientist Singer Nicholson's given the theory of, very important theory given that is the fluid mosaic model there. In here, phospholipids and proteins are present in particular structure. Protein are two types, peripheral protein and integral protein. And next we discuss about the different function of the membrane, that function of the transportation. What is the different type of transport, like passive transport or active transport? This we discuss. We just recall of this part because this is most important, and also we just continue from that part. Cell membrane, right? The simple diffusion, osmosis, passive transport process. Under the passive transport process, why? Because they do not need the ATP or energy expenditure. After that we discuss about the cell wall. You know the cell wall of algae, their chemical composition, cellulose, galactans, mannose and mineral like calcium carbonate. On the other hand, plants consist of mainly the cellulose, hemicellulose, pectin and the proteins molecules. Very important information. Next we discuss about the structure of the cell wall, primary cell wall, secondary cell wall and middle lamella after we discuss the function of the cell wall. Now today we discuss the most important part that is called the endomembrane system. So why is named like this? Because it is a group of membranous organelles having considered or coordinated their function. Their function are correlated. They are all depend on each other for their function. So they are called endomembrane system. In the endomembrane system, like they are the four friends, right? They work together to help the cell. What is the four organelles is there in the endomembrane system? Endoplasmic reticulum, Golgi complex, lysosomes and the fat walls. Now today we discuss one by one. At first, the endomembrane system, it is a or endoplasmic reticulum. You know, this is ultimately a tiny, just tubular ultimate structure scattered into the cytoplasm. Here ultimately, if we ultimately divide or classify the ear, that time it is the two type. That means one intercellular space into two compartments, that is called the luminal inside the ear and also the extranuclear cytoplasm. That means that ear in the intracellular space, it is divided into the two parts. Luminal means inside the ear, like inside this ear portion, inside that is called luminal part. And this one, this cytoplasm part, it is called the extraluminal portion. And the ear ultimately two types according to the attachment of ribosome, rough endoplasmic reticulum and smooth endoplasmic reticulum. In here, what is the difference between the R ear and the S ear? You know, the R ear, rough endoplasmic reticulum, that means they have the ribosome on their surface of the structure. Now, R ear frequently observed in the cell's activity involved in the protein synthesis. So, also the R ear ultimately function is the protein synthesis and the secretion, two most important functions. Also important, they are extensive and continuous with the outer membrane of the nucleus. Very important question. From this outer membrane part, R ear are like developed, right? So which one is the near? R ear and S ear from the nucleus. R ear at the nearby side is present there compared to smooth endoplasmic reticulum. You know, the smooth endoplasmic reticulum, that means the ribosome are not present there. So what is their function? Their most important function like the lipid synthesis. In animal cells, lipid-like steroid hormones are synthesized here. So two important functions, lipid synthesis and lipid-like steroid hormone also is synthesis from that smooth endoplasmic reticulum. Also another function is there, that is called the detoxification. That is the most important function of the ACR. Clear? This function is very important in your NEET part. Now we discuss about the Golgi complex. Also it is called the Golgi apparatus. Why? Because this is ultimately densely extended reticular structure near the nucleus. Why near the nucleus? Because they are functional associated with the endoplasmic reticulum, so they are generally found near to nucleus. And this new Golgi complex is first observed by the Camillo Golgi 1898. From that, this ultimately scientist name, we given this name the Golgi complex or Golgi apparatus, because this is not a single structure. They have the combination of different structural form. In here, you know, they consist of flat disc-like structure that is called the cisternae. This one is the cisternae. What is their diameter? 0.5 to 1 micrometer in diameter. These are stacked parallel to each other, right? Cisternae in here are ultimately concentrically arranged with the convex and concave phase. Convex means the seas or forming phase. That means when the endoplasmic reticulum form or protein, that time these vesicles are receive this thing. So that is called the forming or seas phase. After that, in here the proteins are modified and packaged. After that, from this particular region, this protein, modified protein, are exit from that particular parts. So that is called the trans phase. But in here, bear in mind, cis is called the forming phase. That is ultimately present near to endoplasmic reticulum. It is the far side, away from the nucleus. And the trans or maturing phase, cis and trans phase are totally different but interconnected. Right? You understand these two terms, the cis and the trans. Next we discuss the most important functions of the Golgi complex. You know, the Golgi complex ultimately secrete the material, the intracellular ultimately targets. In the inside the cytoplasms, they ultimately secrete some materials. Also the outside of the cell. If you know, like pepsin enzyme, it is required in the stomach in your body, but it is ultimately where it is from? It is formed in your cells of the stomach. After forming these structural proteins, right, this is just came out from the cell and present in your stomach. Which one is participating in the digestion? So by which cell organelles they are transported from the cell? This is the Golgi apparatus. So at first, you know, the materials be packed here, so it is also called the packaging center. After the packaging here, you know, the endoplasmic reticulums, they just forming a protein, they are received by the cis phase, and after modifications they exit from the trans phase. Also, protein synthesized by the ribosome of the ER are modified in the cisternae of Golgi apparatus before they are released from the trans phase. So also it is the protein modification side. So the secretions, packaging and the modifications is the most important function of the Golgi apparatus. Another two important point is written in the NCERT book. That means they help to formations the glycoproteins and glycolipids. What is it? Glyco means the carbohydrate. Carbohydrate and protein, that is a conjugated protein, and this is the conjugated lipids. That is also formed by the help of Golgi apparatus. Now we discuss the structure of lysosome. Lysosome is a membrane-bound vesicular structure. Why? Because in the lysosome, different enzyme is there, right? So it is ultimately by the process packaging of in the Golgi apparatus also. So lysosome ultimately vesicles, they contain the different types of enzyme. What type of enzyme? The hydrolytic enzyme is there, like the lipase, like the protease, like the carbohydrates. That means the aminolytic type of enzyme. So all the different type of enzyme are present inside the lysosome. So what is their function? But at first you must clearly understand that every enzyme have an optimum pH, optimum temperature. As like same thing is there. They are active at the acidic pH. So all the enzyme in the lysosome, their optimum temperature, optimum pH is acidic for their optimum action, maximum action. So they digest the carbohydrate, protein, lipids and nucleic acid. So you also bear in mind In here, lipase, protease and carbohydrate. But like nucleic acid, which enzyme degrade this thing? The nuclease. So you also tell that nuclease enzyme also be present in the lysosome. Show the 4 different type of enzyme from your NCERT point of view. Next we discuss that the vacuoles. You know they are ultimately membrane bound space where it is present in the cytoplasm. They are contain the water sap because they ultimately contain the water. Also they ultimately contain the excretory material. Also sometimes some gas molecules also be present in the vacuole. But you know the vacuole are covered by a membrane. What is the name of the membrane? The name of the membrane is the tonoplast. What is the name? Tonoplast. And you know in the plant cell the vacuole can occupy up to 90% volume of the cell. Why? Because the plant cell is the site of food production where the food is produced and stored into the vacuole. So the plant cell have a large size of vacuole compared to animal cell. You know the vacuoles in plant cells, tonoplasts help to facilitate the transport of ions and other materials against the concentration gradient into the vacuole. That means low to high concentration. So here the active transport are happening, and here the concentration is higher vacuole than the cytoplasm. Active transport in here. But you know in the amoeba is protistean, right? They have the contractile vacuole, special type of vacuole. What is their function? Contractile vacuole help in the excretion process. What is the function of contractile vacuole? Excretion, help in the excretion, right? So in many cells like the protista, they have also food vacuole. Why food vacuole? Because when they are ultimately engulf some food molecules, they are ultimately stored in a vacuole. That time it is called the food vacuole. In some protistians, you may observe this type of food vacuole. So amoeba also be one type of protista, right? So in protistians, the 2 type of vacuole extra present. That is a contractile vacuole, also the food vacuole. Food vacuole also present in the plant cell. Now we discuss about the very important structure, that is the mitochondria, powerhouse of the cell. So they are clearly visible when it is stained. That means when you are use the dye, after staining you observe the mitochondria. Their number, their shape and size are vary in every cell. You know in our muscle cells the lactic acid are produced because mitochondria number is low. So in our body 2 type of muscle is there, white muscles and the white muscles and the red muscles, because the ultimately their percentage of mitochondria also will differ. So every cells do not have the same number of mitochondria, that is differ to cell. But you know that is ultimately in they are ultimately sausage shape like the cylindrical cell, and they are having the 0.2 to 1 micrometers in diameters and length of 1 to 4.1 micrometer. That information from NCERT, very important information. But you know mitochondria have the double membrane structure, right? 2 membrane you can see it with the outer membrane and inner membrane. You know it is divided by the 2 lumens, outer compartments and the inner compartment matrix. It is called the outer compartments between the 2 membrane, and inside it is called the inner compartment. This is called the matrix. You know, in other words, in that point, that means inner membrane are invaginates towards the matrix. It is called cisternae. This inner matrix and this inner membrane cristae is take part in the cellular respiration. In the matrix, circular DNA material is there, enzyme is there. In here, the Krebs cycle are happening. On the other hand, electron transport system are happening in inner membrane of the mitochondria. So that is the most important side. You know the most important enzyme, ATP synthase, right? These enzymes have the 2 parts, F0 and the F1 portions also been here in the mitochondrial inner membrane, which is help to produce the ATP. Also, you know, many enzymes is there also in the matrix. Also, some circular DNA material is there in the matrix. Also, the ribosome, that is the 70H type. And mitochondria have the ability to self-produce their self-replica by the process of the fission. Fission means the division process. But what is the function of the mitochondria? Mitochondria is the main site of aerobic respiration. Also, it is the site of the ATP productions. So, it is called the powerhouse of the cell, right? So, most important, the important structure and the function. Now, discuss our last topic today, that is a plastid. Plastid are present in the all plant cells and the eukaryotes. Eukaryotes is the protisteans, but they have the but bacterial cells have also the chlorophyll, that is the pigments, but not the plastids. Plastids and the chlorophyll have some different things, because the chlorophyll is a pigment and plastid is a cell organelle which is present only in the eukaryotic cell. You know, their size ultimately easily observed under the microscope. If you do not stain it, but you can observe. Mitochondria do not observe, right? So it is required to staining, but it is easily observed because it is contain the stain or the color. So if they contain some pigments, what is the pigments? According to the pigments, we classify it into the three types: chloroplast, chromoplast, and leucoplast. Chloroplast means green, chromoplast means green, other than green the other colorful plastids, and leucoplast is the colorless plastids. You know, at first we discussed that is a chloroplast. What is that? Chloroplast is the chlorophyll containing chlorophyll and carotenoids pigment. Not only chlorophyll, carotenoid pigment is there. What is their function? They help to trap the sunlight. Their main important function. After that, the photosynthesis are happening. Without chloroplast, photosynthesis do not happening in here, in the plant system. Chromoplast contain fat-soluble carotenoid pigment. One important question: carotenoids, fat-soluble pigments like carotene, xanthophylls. These give a yellow and orange and red color. They also act as accessory pigments that is helping the photosynthesis also. So leucoplasts, they are colorless plastid, but their types are ultimately most important. Amyloplast, they are colorless, but what is their main function? Store the material of food. But what type of food? According to it, we classify the leucoplast. Like the amyloplast, store the starch. In potato, you can see it. And elaioplast, store the oil and fats. On the other hand, aluroplast, store the proteins. That is according to the substance which is there stored, we classify the leucoplast. So this is the three type of plastids that is the most important. And if you are just